Okay, now when you go over to the Google Classroom, I would like you to scroll all the way down. You're going to see a document entitled Scheme of Work. Can everyone see that?
Yes, miss.
Now, this is going to basically go through the topics and the relevant past paper. So since we are going to look for the first topic, which is growth, you're going to scroll all the way down to growth. That is under week 11. There's a past paper. Let us do this past paper. Jan 2016, question 1. So now that you have the question paper, the year, open the paper, let's go through it so you can see how a growth question normally comes. And these are everything you need to know within growth. So you could use this as a guide, this scheme of work document. Go through the topics, you will see all the appropriate subheadings. You will see a past paper attached to it, and you're going to see the diagram that you need to learn to draw and label. So for this topic, you must know the structure, the external and internal structure of the seed. All right, so let's get that going. I'm going to get the paper as well, but I would like you to obviously do it on in your device. Once you get the paper, you can let me know. If you're unable to get the paper, tell me. That will be in a very large document of past papers. Right? If you're using the past papers in soft copy, I always recommend to get it hard copy. There are a lot of places that sell photocopied versions of these past papers. But if you're doing it soft copy, you want to come and click on bookmarks, and you could actually click on the paper itself instead of you just scrolling away. All right, so we go into, what was it, Jan 2016. Right, Jan 2016. And remember, you have the paper trees. People who are private candidates, there are paper trees in this booklet. All right, now the question was the first one, growth. All right, we'll go through it together. So I'm just making sure. Can everyone see question 1?
Yes, miss.
Okay. Now the data provided in table 1 were collected to investigate the growth of plants in light and dark conditions and to make comparisons with the growth patterns in humans. So this is the table here they give you. So they give you the height of plant grown in sunlight in centimeter. Then they give you the height of plant grown in darkness in centimeter. And then they're comparing it now to the growth of a human, well, a human baby in centimeter. So this is something very close to what they are bringing. They say growth in plants versus growth in animals. So you have a time frame from day 5 to day 30, which they recorded the height. For the baby, they recorded the length. So that would be from the head to the toe, they measured. Now, this is the graph that you will have to plot on the grid provided. Now, I'm saying like you have to plot. I don't know. You never know if they bring it in a reverse way. They bring the graph and you do the table. If you have to do a table, you have to make sure you have your columns, you have your rows, you have your headings, and you have your units. And always box it off and use pencil for your lines, right? If you have to do a table. And of course, you put a title. If you have to do a graph, you need to have your label axis. You need to have your scale. You need to have a title on top. You need to have the units in your axis, your label axis. So if it's in centimeter, if it's days, you make sure and include those details. You need to have all plotted points. So what they are asking you, You're plotting the data obtained for both plants. So only plants. Somebody might want to put the human measurements. So they only want both plants on the same graph, right? Using the appropriate scale. And this one, you need a legend. Because you're going to have two kind of information on the graph, you need a legend to show you which point is for the plant in sunlight and which one is for the plant in darkness. So what is going on the X and Y? Who can tell me? The X and Y axis.
Miss, the X will be the measurement and the Y will be the days, miss.
Now you're doing it reverse. Now remember the Y axis is this vertical line here. That's your Y. Your X axis is the horizontal one. So your 1 days, anything to do with time, you always put it in X axis. All right, and then you're putting your height on the Y. So you're basically going to put height of plant grown in sunlight. So let me show you how to construct this. Let me open our next program. Just so you get a little recap with the graph work. Right, so people who are in the drive, you're going into past papers. You're going into paper 2. And you're clicking on this first one. You notice it has from 2005 all the way to 2016. So of course the Jan 2016 paper is here. Then you have from 2017 all the way to 2020, 2021, June 2021 as the latest paper. Right, but right now, before people used to just be doing the latest papers and drilling themselves, but now that we have a guide, CXC giving you the topics, is ready to just master those topics. All right, so and notice 5 marks for the graph. Most graphs is be 5 marks, provided you have everything. So I'll show you how to get it. Now, when they give you that graph paper, you're using the entire graph paper. So your lines have to come in the borders itself. You're using this entire, don't go and draw some small graph. You utilize, you want to show that you understand that you have to utilize graph space, graph page. All right, so I'll kind of make it small here so you could see. Now remember doing it online, so you're you're drawing your lines, like the first step, right? And then you come across with your x-axis here. That's your first thing. Then you come now and you start to label. So I'll just put like a little y here, and then I'll put a x here. Now you have to actually put the actual name. So remember, on the x-axis, you're going to have these. On the y-axis, you're going to have, let's go back and check the table. You want to look at the table here. Height of plant. So you put in height of plant. Right? Of course, you will have more space. Height of plant, and it's measured in, you put the unit centimeter. Now you have to plan your scale. You have to make sure you plan everything. So you look at the readings. You look at the least, okay, what's the smallest reading here?
10.
10, miss.
10. And then you have to go all the way, the greatest reading is 30. So you know you have to start from, well, you could start at 5 if you want, but 10 being the smallest all the way to 30. So you plan ahead. You look at the lines. If you're going 10, let's see, 10, 20, 30, that could fit. You could even space it out if you wanted nice. So you could put like probably, all
Miss
right.
Eki could do it in fives.
You could do it in fives. You could put 5 here, 5, 10, 15, 20, 25, 30. So that will make it go all the way up here. So you plan it as suit, but your scale is important. I'm going to show you how to write your scale. Right. Now, one small block, this small block here is 1 centimeter. So if you're looking at the y-axis, when you're writing your scale, you could put the word scale. Right, I'll zoom in. Right, scale. You could always put your scale in the corner of your page on top. Right. So let's start with the y-axis. What's the scale for the y-axis? So you could do it in fives, and that would have bring it all up here. So 30 would have fit if you're doing it in fives. Now, if you're using 2 centimeter as 5, remember, one small block is 1 centimeter. But if we do it in fives and we put 5 here, 10, 15, that means you have to put 2 centimeter represent 5. So that those 2 blocks represent 5 centimeters. That is the scale you're using, right? In terms of the x-axis, let's get our scale for that one. So the x-axis, they have, let me see how it runs. Them have it 5 all the way to 30. So we just want to make sure it fits in. Always plan your graph. So if you put 5, 10, 15, 20, 30, it stop here. But that graph will be small. You want to use all here. So you could probably do it the same way. 5, 10, 15, 20, 25. Yeah, it will fit with the 2 centimeter. So it'll be the same thing. But when you're doing this scale, you want to make sure you put 2 centimeter represent. Now this one have a different reading. This one is days. So you have to put it represent 5 days. Your scale must be related to what you're plotting, and that's how you do it. And then all you have to do now, you just look at it. So you see the first one is 5, the height is 10, you put your point. So, right, so I'm going to show you the first one. Now, because it's 2 different data, you want to use 2 different points. So the one growing in sunlight, you probably use X's or dots. So I'll just use an X. So let's say this is day 5, and this is 5 centimeter here. You put your point. Right, and you continue doing it. And then you now always connect your line to the 0 mark. You always make sure you put 0 here. You start somewhere. You start at 0. Even though the table didn't have anything with 0, you're connecting your line to 0. So you will just make sure you connect from 0 and do all your points. So it would have been like a graph going upwards, right, throughout the days. It would have been something like that. The next thing, now some points, now when I look at this graph, it's very even. This one, you might have some little indifferences like here. It doesn't mean, now you just have to be careful when you're doing your best fit line. Now this one, it could be a curve. This one could be a curve. So you're just connecting all the points and you're just going into like a curve formation. All right. Growth graphs are normally curves. Now, because you have 2 different data, 2 different information, you want to make sure you have your key or your legend. So you make sure you put legend in the corner, and you put, well, the X's is for plants, and you go back to the table, plants grown in sunlight. And then if you're using like dots, you just put what it represents, plants grown in darkness. And that will give you a full five marks once you have everything in order. Right? And the title cannot just say graph showing information or graph showing measurements. Your title must be detailed. So the title should be something related to every single heading here. So you could have graph showing the height of plant grown in sunlight and in darkness for a period of 5 to 30 days. You want to tie in everything together in the title, right? And that is where the table will help you form your title. And if you're totally lost, you could just change up this. You could just say graph showing the growth of plants in light and dark conditions over a period of 30 days. Right? Remember, you didn't do anything with the baby, so leave out that part. Everybody go with the graph?
Yes, miss.
All right, moving on. Now they are going to ask you something to do with describing the pattern of growth. Now when you look at the information, all of them showed an increase over the 30 day. So this one went from 10 centimeter to 30. This one, however, it didn't grow as fast. It had some growth, but if you notice, it stopped growing at day 15. At day 15, it went to 17 centimeter and it stayed at 17 centimeter up to day 20. Then you see it started to shrink. The height of the plant, it started to get less. So that has to do with the condition it was placed in. So whenever you're doing the pattern, you want to draw a pattern between the two. So you want to say that the plant, the height of the plant grown in sunlight showed how to say, a greater increase over the 30-day period, whereas the height of the plant grown in darkness had a shorter increase and it stopped at day 15. You want to just compare the two in a sentence. That is the pattern, right? Because they say for each plant. And that is 2 marks. So you be careful with your words. Always remember to show the comparison, say which one have a faster growth rate, slower growth. You can say growth, you can say it in a rate, like fast growth rate, slow growth rate, have a greater height, have a shorter height, right? Terms like that. Second part, explain the differences observed in the growth of plants under these two conditions. So this is where the theory will come in. What it is about the light that will have a greater height of the plants compared to the plants that are grown in darkness. So you start to talk about phototropism, you start to talk about how the meristems, the auxins in the shoot is stimulated by light, which will cause it to grow upwards. Whereas in darkness, the auxin is not stimulated. So therefore, it's not going to have that increase in growth or increase in height because the auxin is not stimulated, right? So you put your theory there. And this one is 4 marks, so you need to really properly explain it. I will have to see how you write as well, just to give you a little critique on how you answer any questions. Part C. Complete the following definition for growth by writing the correct words from the list in brackets. So growth may be defined as this is a fill in the blank. So growth is something that is what? What is the best word here?
Sorry, Miss, can you repeat?
What word will come into these blank spaces?
Miss, will it be permanent organs?
Permanent, right? Growth may be defined as a permanent increase in
Organs, Miss?
the mass, the mass and volume of an organism when food is absorbed and converted to living matter.
Okay, Miss.
All right, so it will be mass here. Right, the next set of questions dealt with suggest why the measurements of length taken for the plant are incomplete, unlike the measurements taken for a baby. So remember the little table in the notes. You remember for plants, let me go back to here. Plants can regenerate quite extensively and growth continues throughout life. Whereas in animals growth stops at adulthood. So the measurements of the length taken for plants are incomplete because it could always extend. Whereas the measurements taken for the baby, well, you have a cut. Well, the baby will obviously have a cutoff point from baby stage, that toddler, then it have young adults or adolescents, and then will they have adulthood. So it will be different for these two. Then they ask you to suggest 2 ways in which this experiment could be refined to obtain more accurate data for measuring growth. So anytime they have something to do with how you could change the method or how you could improve the experiment, you could always talk about you prolong the experiment longer. Instead of just 30 days, you could do it for about 2 months. So this is just, let's say this is 1 month. You could do it for 2 additional months where you record the height. So you get accurate growth measurements. You get accurate readings, right? And also you can have more than one plants in sunlight, more than one plants in darkness. Instead of just counting from one particular plant, you can have multiple plants where you measure. That will give you a better idea of the growth. So that is some ways you could improve our experiment, prolonging it, increasing the number, right? So what I would invite you to do now, I'm just going through the past paper right now. You will be given time that you will be doing. I would like you to do your own answers, and I will create I'll create a portal for you. So you see all the individual past papers pertaining to the topics that are coming for CXE. Like for instance, growth, you will do out your answers for this past paper. The next topic on the list is germination. So when you scroll to germination, now germination will fall under I don't want to give you a wrong, okay. Germination would have been under growth, all right? So I will have to find a particular question based on the germination process. I will let you know what year. The next topic is autotrophic and heterotrophic, so that is nutrition. You go to the topics, right? Animal nutrition, plant nutrition. You do all the papers that you see and attach to it. So you get an overall revision of those topics. Now, you're free to send me your answers if you want me to just look at it and tell you how you're doing, right? How you're going in it. If there's changes to be made. All right, for each 8 topics that CXE sent. Right, so it was very important that I go through the graph again with you so that you don't miss out anything. Let's move on to another topic. Right, so I'm just going through now since photosynthesis You never know if they're asking something pertaining to the leaf because the leaf is part of photosynthesis, right? There's the organ that is involved in the process, so you never know what they bring in it. All right, let's make this full screen. Can you all see summary of parts of internal structure of the leaf?
Yes, miss.
Okay, great. Now each part of the leaf is important. This is the internal view of the leaf. So you always start with the top part or the surface, which is the upper epidermis. So you see how they magnify that section of the leaf? This is it in a cross-section view. So you have the upper epidermis, you have your palisade layer, you have your mesophyll or spongy tissue here, spongy mesophyll cells, a lot of air spaces here, and then you have your lower epidermis. So each part I have a detailed function. So the cuticle is on the upper. That is like a waxy layer. It's waterproof. It is secreted by cells of the upper. So when light is entering the plant, you don't want too much light entering because it can damage the plant. So that waxy layer does actually cause some light rays to bounce off. All right, it's not like all the sunlight being absorbed, just a small amount. The upper epidermis are these cells which are thin, transparent, meaning light can pass through. There's no chloroplast here. They act like a barrier to any disease organisms. So it prevents the plant from getting disease. The palisade is important because if you look at these cells, it's a rectangular shape. This is where you have your chloroplast. So this is the main region for photosynthesis. So if they ever ask you where photosynthesis is take place, it's in the palisade layer. And spongy mesophyll, now they tend to be more spherical, loosely packed. There's a lot of spaces. They contain chloroplasts, but not as many as the palisade. The air spaces in between does allow the gas to pass through or diffuse into the leaf. Now gases come from below. So between the lower epidermis, you're going to notice it have stomata. Stomata is little tiny holes or openings. So you're going to have carbon dioxide passing through, water leaving during photosynthesis. All right, and the opposite for respiration. So diffusing all through the air spaces till it comes into the palisade layer here. When it reaches the palisade layer, then the gas will react in the dark reaction, making food. The vascular bundle is situated right in the spongy mesophyll. You're going to have a group of cells forming the xylem and the phloem. Right, that is like the vein of the leaf. The xylem is to bring water and minerals. Phloem is to transport the food that was made. Right, so that's basically the parts of the internal structure of the leaf. Right, the next topic you had to learn was the plant and animal cell. So they're of course going to ask you to compare the two. Right, the plant cell, it have a cell wall, animal cell don't have any. It also have one large vacuole that is permanent. This is the white area, there's the vacuole. Animal cells tend to have small numerous vacuoles. They both have a nucleus, they both have cell membrane. Remember, the membrane has the little pores. Cell wall is permeable, anything can pass through, but cell membrane is allows certain things to enter the plant. Right, now if you notice, this one showing you which one have the organ. Animal and plant cells have these. Plant cells alone have the cell wall, the chloroplast. Right, and then we'll have the functions for each part. Chloroplast is the organelle that photosynthesis takes place. Remember that. It contains the green pigment. The cytoplasm is basically the jelly-like medium that surrounds the cell. That is where you're going to have, well, substances becoming dissolved, being transported around the cell. And well, the nucleus is important because that is the brain of the cell. You have DNA, you have your genes, you have all your genetic information stored, and genes govern who you are. Genes controls everything in the body. So that's why you call it the brain of the cell. So look at how much things it controls. Cell division, cell development, cell everything that's going on in the cell is controlled by the nucleus. So this is a little summary table showing you the comparison of the two cells to learn and label. Right? Make sure you go through diagrams. They tied in osmosis with this topic. So not talking like I saw the paper, but remember, I just go in with the order of the list of topics. Osmosis is when water moves from a region of high concentration to a region of low concentration. So whenever you put up, let's say like a plant cell in fresh water is pure water. It have no salt, it have nothing dissolved in it. So you're going to have water entering the plant, right? Because they're moving from a high area to a low area of concentration. So inside the plant cell is lower than outside. So the plant cell is going to become turgid, meaning it's going to swell. So notice the arrow showing where water entering from. If it's in a balanced solution, meaning the water that it placed in is the same contents as the water that is inside the cell, you're not going to have too much. The same amount of water leaving will be the same amount of water entering. So it's like a balanced solution. So it remains flaccid. If you put it in salt water, all of the water in the plant is going to leave. So you see how water leaving the plant? It moves from an area of high concentration, which was inside of the plant, to the region of low, which is outside. Salt water means it hardly have water. It have more salt than water, right? And you notice all of the cytoplasm shrink. That is when you have something called plasmolysis. The cell become plasmolized. So the cell, the cytoplasm, strip away from the cell wall, right? So this is what occurs when you put a plant cell in salt water for a long period. All right, before it becomes plasmolized, it would become flaccid first. But then when all of the water leaves, it becomes plasmolized. These are just different kinds of solutions. So you call this hypotonic, isotonic when it balanced, hypertonic when plenty water leaves. Right? When water entering is hypotonic. So those are terms that you just want to learn. You want to make sure you understand these two terms. What is concentrated and what is dilute solution. So look at the figure. Dilute means when there's a small amount of solute. Solute is the little solids. So if like for instance, weak coffee, if you hardly put any coffee in some water, you could call it dilute because it still have more water. It have more water than solute. Concentrated is when it have more solute than water. So like strong coffee, if you just have more coffee and less water, that is concentrated. So anytime you have a concentrated solution, water will always be going towards that kind, right? Because osmosis is always when water moves from high concentration to a region of low. If you have a dilute solution, water will always leave the dilute and go to the concentrated. That is just the movement of water, right? In the body, in plants, you see osmosis taking place in the roots. You see osmosis taking place when you, like for instance, when you drink water and water have to pass through your digestive system. That is how it will move by osmosis. So those are terms to be familiar with. Make sure and learn it. Any questions, by the way, with cells and osmosis?
No, miss.
No, miss.
The next thing with cell is cell specialization. So some textbooks, they have it as cell differentiation as well. It just simply means you have multiple cells that are specialized or they are just special in what they do. So cell specialization, multicellular organisms have millions of cells and they have different kinds of cells. So each cell has a particular shape, size. They all tend to have the same structures like nucleus and cytoplasm. Who can tell me the name of the cell?
Um, pretty sure, I'm not too sure, but is it the yes, but I think it's the reflex
No, I say what kind of cell, not
kind of cell. No, miss. I don't really know.
there's a nerve cell, the nerve cell.
Yes, that was it. I said reflex, but I also say nerve. That was it.
Yeah, nerve cell. Right? So you remember you have to know, well, definitely when you're looking at the listing of the topics, they say blood components. So you have to know what a red blood cells look like, huh? And your plant cell and your animal cell. Okay. Now basically the word specialization, it means that multicellular organisms have more than one cell. Multi means many. Each type of cell is different in size, shape, and structure. And the reason for having different size, shape, and structure is because each cell have to perform their individual task effectively. Right? So an egg cell have to, the purpose of an egg cell is to become fertilized. The purpose of a nerve cell is to transmit impulse. Purpose of the red blood cell is to transport oxygen. So they all going to have a particular shape and size to facilitate its function. Right? So to perform their task effectively, cells will go through differentiation process and specialization. For example, nerve cells have thin, long fibers called axons, which conduct nerve impulse. Right? So that is what cell specialization is about. Right, so let's look at a question pertaining to 1, 2, 3. Right, something on cells. All of that will fall under cells. So you go to your scheme of work, you go to cells. Let's look at this Jan 2017. Question 2. So you open, right? You open your Google Drive, look for Jan. Well, these easy to find. You go to question 2. All right, so let's take a look. So let me see. I'll ask someone to read and give me an answer. You know, we could do it orally as well. All right, so Alex, go ahead with question 2A, part 1.
State three differences between a plant cell and an animal cell. A plant cell has a cell wall, an animal cell doesn't. The plant cell has chloroplast present, and the animal cell has no chloroplast present.
One more.
The plant cell has a rigid cell wall and the plasma membrane, and the animal cell has a flexible but thin plasma membrane.
No, we can't say that. Both of them have a cell membrane. what you have to say is the plant cell have a large permanent vacuole and the animal cell have small numerous temporary vacuoles. All right,
Okay.
so write that down somewhere. Next question, part 2. Let's hear from Ambika.
Part two.
Yeah,
uh, the shape of
read
it.
the question.
Say two differences between a prokaryotic and a eukaryotic cell. Um, the DNA of a prokaryotic cell is circular and free, while the DNA of a eukaryotic cell is enclosed in a nuclear membrane, and the prokaryotic cell contains no membrane-bound organelles, while the eukaryotic cells contain membrane-bound organelles, such as the mitochondria and the chloroplast.
Very good. That's an excellent answer. That will gain you 2 marks, right? Now, those are categories of cells. Prokaryotic cells is like a bacteria. Eukaryotic cells would be like your plant and animal cells. So you just think of how the bacteria cell is. Bacteria have no nucleus. You could also say it have a tail. Eukaryotic cell have no tail. And just as she said, the prokaryotes, they tend to have the structures in the cell don't have a membrane. Whereas in eukaryotes, that's your plant and animal cells, remember that? All of the organelles have like a little barrier. So the chloroplast have a membrane, the vacuole have a membrane, right? Things in the prokaryote just floating in the cytoplasm. It have no particular barrier. And there are many things you can mention too. So I would basically give you a little, like a table differentiating these two on Google Classroom. I'll post something for you. All right, let's get somebody else for part B. I have, okay, Jaden, part B.
Now, it's the function of each of the following cell wall. So the cell wall, it protects the cell and prevents it, well, from any damage. It also gives structure to the cell and it prevents it from wasting like the animal cell with only a membrane.
Mm-hmm.
And the cell membrane, well, it's also it's a semi-permeable shell membrane. It's a semi-permeable membrane, right? That protects the interior of the cell from the outside environment.
No, when you talk about protection, talk about cell wall. When you talk
Cell.
about membrane, you talk about it controls what enter and exit the cell.
Okay.
Right? So cell membrane is partially permeable. The cell wall is actually fully permeable. Anything can pass through that cell wall. What a cell wall is doing, it does protect the cell, but it also prevents it from bursting, as he said, and it also supports the plant. Because it is made up of cellulose, that's a tough material, it supports the plant, it keeps it upright as well. All right, so good so far, cell membrane. Right, this one have to do with osmosis and stuff. So let's see. Let's hear from Anushka Patsi.
Explain what happens to an animal cell when it is removed from the organism and placed into concentrated sucrose solution. The animal cell would become shriveled because the water would leave the cell.
All right. So now animal cell when removed from the organism place. So the key word here is concentrated. If you're putting an animal cell in a concentrated sucrose solution, remember concentrated means less water and it has more sucrose in it. So the water from the animal cell, the water from the cytoplasm and the vacuole have water to it. It will leave the animal cell. So it is going to become trivial. As she said, you want to use some biology terms here. So you want to use the word flaccid. You could use the word flaccid here. All right. Next one, part 2. Someone else. Jazeera, part 2. All right, I get a bit static, but it will just be the opposite. So look at the word, it is placed in distilled water is pure water. Distillate, water that has been distilled means all impurities were removed by the process of distillation. So that is pure water. So if it is you put an animal cell in pure water, that animal cell going on swell. And basically the water from the distilled water, that is a high concentration compared to the water in the animal cell. So it enters the animal cell. So it becomes turgid. Right? Use the word turgid. All right. So the last one here, I will go through this one. Describe how describe how the results would be different from C, part 1 and 2, for a plant cell in concentrated sucrose solution and distilled water. So you're just explaining everything, but in a plant. So you have to consider that cell wall. Now in concentrated sucrose solution, if you put a plant cell, yes, the water from the plant cell will leave the plant and enter the concentrated sucrose solution, right? And if you leave that for long, remember the word I mentioned, it becomes plasmolyzed. Plasmolyzed means that the cytoplasm stripped away from the cell wall. All right, so that is how it will be different from the animal, because the animal don't have a cell wall. If you put the plant cell in distilled water, now the difference here, it will become turgid, but it will not burst. It will not burst because of the cell wall holding the contents. If you have the animal cell in distilled water, eventually it bursts. It becomes turgid and then it bursts. So you want to put that for 2 marks. When you put in the plant cell reaction, you're making sure and saying it will become turgid because water will enter the cell, and eventually it will not burst due to the presence of the cell wall. All right, because you want to gain your marks, you want to be thorough in your answers to show the complete difference between the two, complete difference between the plant cell and the animal cell. So that's something that could come, something close, something change it up in a way that instead of the same concentrated solution, they could probably say concentrated glucose. It means the same thing. They could just say salt water, something. Look out for those keywords. That's the idea. Okay, so let's move on to another topic. All right, we finished with cells. Now we're moving on to our environmental topic now. So they mentioned that they want to bring the effects of climate change and they're paying attention to the conservation of the environment. Now, first off, what does cause climate change? What does cause the change in the climate? And climate can mean many things. Climate can mean weather, temperature, right? Climate is not, it's multiple things. So as you can see, there are various causes. Rapid industrialization. So when you have, let's say, the cutting down of trees and the building of industries and companies, you're going to have more air pollution. Energy use. We rely on fossil fuels for energy, and that has shown to cause a lot of air pollution, particularly carbon dioxide. Agricultural practices. Some methods are very damaging to the environment, like slash and burn. Um, Sometimes they remove when they are cutting down trees for agriculture, the soil becomes, stripped of certain minerals. Also, when they're using a lot of heavy chemicals, pesticides, fertilizers, those things could also pollute the environment. So pollution is a cause of climate change. That is the baseline here. Um, Transport, livestock, okay. So everything here will be basically emit some sort of toxic gas or some kind of chemical that can alter the climate. All right, so you have to make sure you learn your greenhouse gases. You have several greenhouse gas. You have, for instance, carbon dioxide, nitrogen dioxide, sulfur dioxide, CFCs. Those are contributors to climate change. Now, what are the effects of climate change? You are going to hear the word global warming. Global warming is when the entire Earth temperature has risen. And when that occurs, which is, it took place, you have a whole, it increased by 1 degree. I can't remember the exact thing. It increased by 1 degree, which is a lot. That is going to have several effects on the Earth. So not only does the temperature rise, sea level does rise. When those polar countries, all the ice caps melt, it will cause the sea level to rise, which will lead to flooding. You're going to have unpredictable weather patterns. So you no longer have like a set season, rain and rain season, dry season. It's, sometimes it bright, it's sunny out, and then all of a sudden it could just turn dark and gloomy. There's an increase in extreme weather events. So you might have hurricanes, you tend to have earthquakes, volcanoes, a lot of extreme weather events or environmental disasters, land degradation, loss of wildlife and biodiversity. Because of that sudden change in the climate or the extreme temperatures, wildlife couldn't adapt it. There will be a lot of deaths. A lot of the plants and animals couldn't survive that extreme change. So you will have some loss of biodiversity. All right. Let's look at the conservation of the environment. So what are ways you can implement to save the environment? Conserve means to save. So instead of having a lot of vehicles on the roads, a lot of carbon dioxide being emitted from these exhausts, and not only carbon dioxide is being emitted, you have sulfur dioxide, nitrogen dioxide. You could carpool, you could use a bike, you could walk. This is important for the air pollution. You will always have these 3 R's. These 3 R's are important when you're explaining this kind of question. Reduce, reuse, and recycle. And you must know the difference. So let's start with reduce. Reduce means you cut down on your plastic, glass, paper, what else? Plastic, glass, right. Instead, like for instance, there are a lot of groceries that have that made it mandatory. You have to come with your own bag, right? They don't, or you have to buy their bag. Some policies like that. So that will reduce the amount of plastic. You have reuse. Reuse, like if you have a plastic bottle, you refill it, right? Instead of throwing it away and buying another bottle, you just reuse it. So that cut down on the amount of plastic waste. Then you have recycle. So instead of discarding a material, a container, a bottle, whatever you buy, you could change it up. Instead of now you're reinventing a way, you could use that apparatus, that material in a different way. So I always tend to use this example. When you buy those large plastic bottles, those water bottles, some people is change it into a plant pot. They simply bore some holes. So that is recycle. All right. Make sure you know the difference of the three and make sure you can give an example. Now, they have other ways, like for instance, reforestation projects, planting trees. You have they ban certain aerosols that contain AFCs, CFCs. You could choose non-toxic chemicals, so they are biodegradable chemicals. They are actual cleaning agents you could use that are natural. Instead of buy less plastic, bring reusable shopping bag. Okay. Then they had this energy conservation one. Instead of using the yellow bulb, they use the LED bulbs instead. They are energy efficient. Take off the lights as well when not in use. You can conserve water by turning off the tap. When you're brushing your teeth, use a cup. Fill a cup with water and use that alone. There are many different ways you could talk about it. Depends on the question, right? It's always really a question. Educating yourself, educating others as well can help to conserve the environment too. Volunteering to clean up in your community, getting involved in protecting your watershed too. All right, so watershed is like a water reservoir where water is contained. All right, so this shouldn't be too difficult. And I know it's a lot of environmental parts here. Any questions, by the way? If I move to the next topic.
No, miss.
All right, so we will do the blood components. All right, and then we'll do a past paper on those two. And then we have to take our lunch. Lunch is important. You need to fuel yourself with energy. You need a break away from the screen. Like I said, at this point in time, your phones need to have limited screen time. There's a lot of widgets or apps you could have to show how much time you spend on your phone. Anytime you see like it exceeds 30 minutes, you know it's time to put it down. Your phone should just be a tool like emergency purposes. I wouldn't tell you to come off social media. I'm not going to be like, oh, come off and deactivate. I know you're not going to listen, but remove the app from the phone. From now till June, remove those apps. You're still in it. You're not going anywhere. It's just dormant for now. You are going to be wasting a lot of time scrolling through these apps. So it's advised you take it off from the phone. All right. So you have limited screen time on your device. All right. So I'm going to move into blood components. Now there are various components is the cells or what makes up blood. Blood, remember, is a medium. Blood is a tissue. So although blood is in a liquid form, you're going to have the various components. The majority of your blood is actually plasma. That is the liquid watery medium. So it's about 55% of your blood. You need it to dissolve nutrients, transport hormones, right? Red blood cells, now this amounts to about 40 to 45%. That's an important part of the blood. It carries oxygen throughout the body and it helps to remove carbon dioxide. Your white blood cells, now these are part of your immune system. So you're going to have your two types of white blood cells, phagocytes, lymphocytes. Phagocytes contain a lobed nucleus and engulfs and digests pathogens. Lymphocytes contain a round nucleus. So look at the difference. One is lobe, one is round. And look at how they act. Lymphocytes produce antibodies to neutralize antigens that are produced by pathogens. So your white blood cells now they make up less than 1% of the blood. All right, so remember their purpose is to detect and fight viruses and bacteria. Basically prevent an infection from forming and prevent a disease from happening in the body. Platelets is when you get a cut or a wound that is going to seal the wound and stop excessive blood loss and also seal it so that foreign pathogens cannot enter the body. And they make up about 1% of blood. Right? So if they ask you to draw a red blood cell, remember it's a biconcave disc shape. The white blood cell, it have a, well, one is lobe nucleus, one is round. Platelets is basically fragments of cells, so there's no nucleus. Red blood cell have no nucleus as well. Right, so those are some points to also take into consideration. Sickle cell anemia. So this is a genetic disease. This is meaning you can inherit this disease. So what is sickle cell anemia? Now you have two kinds of anemia. You have sickle cell and you have iron deficiency. The iron deficiency one is you're just lacking iron in your diet. Sickle cell anemia, you're born with abnormal red blood cells. So the abnormal shape limits the amount of oxygen flowing in the body because there's less surface area. So look at how normally it's supposed to be concave, biconcave disc shape. When it's this crescent shape, it tends to clump up easily. So you see, if it's biconcave, it can, move freely. But because of this appearance here, it tends to kind of form clumps in the body, and that can lead to death if the person have the disease full blown. Now, the disease is actually come in handy for persons who live in a mosquito-borne environment. So areas that have a lot of mosquitoes that spread malaria, you have a higher chance of surviving if you have sickle cell disease. And that is simply because of this crescent shape, the virus cannot reproduce in that abnormal shape. It could destroy and attack the red blood cell, the normal red blood cell, but not this crescent shape one. So it have an advantage, all right? That's something when you really read a textbook, you'll get into those kind of information. So we have to look at that genetic, before I go into menstrual cycle and stuff after lunch, we have to look at how the genetic question can come for sickle cell anemia. So I'm going to get that question for you, right? All right, so I will post up the same question for you to practice, but I'll go through it first. All right, so let me show you. All right, I'll post it in the Google Classroom as well. Right, I'll entitle sickle cell genetic question. All right, so you can look at my screen from here. Now, we have to be familiar with how to use the particular codes. For genetics, you use symbols or you use codes. Now, for sickle cell, there is a numerous ways. Sometimes they could just give you like a one letter, or sometimes they will give you like the protein and the allele. Now, you have to know these terms as well. So I'll use this question here as an example, and I'll get an ordinary one with just the simple allele symbols. Don't get confused with the alleles and the symbols. You just have to know what certain terms mean. So sickle cell anemia is an inherited disease, which we know. The gene for hemoglobin exists in two forms. You have now HB stands for hemoglobin. That is the iron molecule in your red blood cells. So that is why they put this HB here. So the allele is normally attached to that protein, that hemoglobin protein. So don't get confused when you see HB. We are more concerned with these letters here. So they have N, which would be for normal, and then they have S, which means the person has sickle cell disease. So they're telling you person who have SS, right, have the disease and experience symptoms including fatigue, meaning feeling weak, extreme pain in their joints. People who are NS, so when you have the normal red blood cell, together with the sickle cell, you are what you call a carrier of the disease. So you may have mild symptoms or you mightn't have none at all. So you could still live a normal life, healthy, right? You might still feel weak here and there, but not deadly, as if you have the actual disease full blown, meaning you have the two alleles being sickle cell. Now an allele, you have to complete this table here by stating a specific example using the paragraph above. So the genetic term and the example used in the passage, allele. Now when you look at this passage here, the allele is what is given in the symbols, in these letters here. So when they say, now an allele is made up, a gene is made up of two alleles. So when they say a gene for hemoglobin exists in two forms, the two forms that they are talking about is these two alleles. So that would have been the example here. So you're basically filling in HB capital N, HB capital S for the allele. A heterozygous genotype. Heterozygous means different alleles. So that's when you have two different alleles paired together. So between these two, which one have different alleles paired together? Anybody?
Let's repeat the question, please.
Between these two symbols, which one have different alleles?
Okay, so between two symbols have different alleles. That
Yes.
wouldn't that be the carrier? Because you have HBN and then HBS.
Right, because it have NS, that is two different alleles. This one have SS, that is the same allele. So because these are N and S is different alleles, so that is what you call a heterozygous genotype. So you're basically putting these symbols here under this row. Homozygous. So hetero means different, homo means same. Right? Same. Homozygous, with the one with the same allele, would be this one here. The one with the 2S. Alright. Now, anytime you hear the word genotype, they want the symbols. So symbols could be in a heterozygous form, or it could be in a homozygous form. Hetero means different, homo means the same. If they mention phenotype, they're talking about phenotype means physical. Physical, what could be seen or what exists. So the phenotype in this passage would be, now when you read the passage thoroughly, they say people who have SS have the disease and experience symptoms including fatigue, extreme pain in the joints. So that is something that is expressed from the gene. So if you have this gene, your phenotype will be you have the disease and you experience symptoms including fatigue and extreme pain. Or if you have the heterozygous form, or if you are a carrier, then you may have mild symptoms or if any at all. So these two sentences will come under the example used in the passage for phenotype. All right, so I have some concise notes that goes through these terms. I'll post it up in a bit. All right, so you get an idea what it means. And that is just to fill in a table. This question have to talking about that same thing with malaria. Want you to practice that. So I post up the question here, right? But we still have the genetic diagram to go through, right? If we have a question with a sickle cell case, we have to know how to work it out. So I'm going to get at that particular question. So when you go to your scheme of work, you're going straight down to genetics. Genetics start from, all right, so it'll be somewhere here under week 16 where you have use Punnett square. So there are two genetic diagrams. There's the Punnett square and then there's the test cross. I'll start showing you the Punnett square first. So let's go over to this paper, Jan 2019, question 3. Jan 2019, right, that's the same paper. Right, and we scroll into question 3 now. Right, this is question 3. And this is blood groups. All right. All right, I particularly want a question on sickle cell, so I'll try this one, 2018 instead. Now, feel free to work out those others as practice. Huh? Right, 2018, question 6C. Let's see. So that's all the way to the bottom. 5. Okay. This is a sickle cell question. No, this is hemophilia. Hemophilia is another type of disease, right? But I want to make sure we're doing something on sickle cell. Jan 2017. 3C. Hemophilia again. So they bring hemophilia so many times that they decided to bring sickle cell. All right, I'll try this 2016 one. I know somewhere in that it will have a sickle cell question. Eh? All right, question 5B. Let's see. 5B, Jan 2016. All right, so here is a sickle cell question. I'm going to cut off this question. Now, in the beginning of this question, look at how they ask. They say name 4 major components that make up human blood and explain how the oxygen transported by the blood becomes available to cells for the production of energy. Look at how similar in terms of the topic. So they're talking about blood components, and then they're talking about sickle cell. So this is something very close to what could come. Right, I'm going to cut this part off. This one is 2 marks, then this one is 5 marks. Now, when I pose these questions, it means you got to do it and send me some answers so I could see how you're writing the answers. But this one, I'll work it out for you so you can see the steps. Right, sickle cell disease genetic cross. Right, so the first part of the question, sickle cell anemia is an inherited condition in which the blood of affected persons cannot transport oxygen efficiently. So we know the theory. Can everyone see the question?
Yes, miss.
Part 1. Describe 2 symptoms of sickle cell anemia. So you could say person will feel fatigue, they can get pain in their joints, the list goes on. All right, they feel weak, tired easily. Part 2 is where you have your diagram and you have a lot of steps to earn that 5 marks. So let's get someone reading this one. I'll call on, so I have to make sure people listening and focusing. I'll call on Kyle.
Fazul and Shana both carried the sickle cell trait. What is the chance that a child born to them would suffer from sickle cell anemia? Explain your answer with the aid of a genetic diagram. Use the following symbol A, which is normal, and S, which is sickle cell.
Very good. Now, when you look at the previous question, now there is 2 ways they could bring these symbols. They could bring it with the hemoglobin attached, right? Or they could bring it like in this question here, just using the letters. And when you use the letters alone, it's much easier to work out. It doesn't confuse you when you're working. Now look at what the symbols stand. Capital A means normal, capital S means sickle cell. The previous one was the difference, it was capital N for normal. Right, but we have to use this one. Now, the key to the question is understanding who's the parents, right? They say that Fazul and Shana both carry sickle cell trait. So, does this mean that they have the disease or they are carriers? What does that sentence mean? Who say they are carriers? Raise your hand.
I believe they are carriers.
Yeah, they are carriers. Excellent. They say they both carry the sickle cell trait. They didn't say they're both suffering from sickle cell anemia. Now look at the second sentence. What is the chance that a child born to them would suffer from sickle cell anemia? Now, do you think if your parents are carriers to the disease, can you inherit the disease?
Yes, we can.
Yes, we can. Yeah, it's possible. So now you have to show using a diagram. So I'll show you on screen. The other key to that question, they ask you, what is the chance? The chance, you have to give it as a percentage, a ratio, a fraction. What is the chance? Right? So we know that it's possible, but what is the chances? Is it 100% chance that yes, that child will not have the disease? Is it 50% chance? Okay, the child could be normal, the child could have the disease. So we have to use the diagram to get the chance. That is the whole point of having a genetic diagram, understanding the chances. So when you're working out this, you must have some, okay, so let me see how to split this screen. You all still seeing?
Yes, Mrs. Nico.
You're supposed to see this and right, can you see the past paper side by side with a blackboard, a whiteboard?
No, miss, right now I'm just seeing a white screen.
White, all right. Right, I'm just getting back the question. So if you want to refer to the question, remember it's Jan 2016 question 5. Right, so what we're concerned about is how to work out this. Now, like I said, to get the 5 marks, you need to have some lines. You can't just draw a square and just do the working anyhow. In your paper 2 is all about earning marks. In your paper 1, well, it's just about getting the right multiple choice, the right answer. But the paper 2 is you're writing for your answer. So what you want to start off saying is, you want to say who's the parents. So you want to put the parental, you'll see in parental, right now.
Yes, miss.
Now, when you start off with the parental phenotype, the phenotype is given in the question. And according to the question, they say the both parents are the carrying the sickle cell trait. So you don't have to go and write out this whole thing. You're just simply saying the parents are carrier mating with a carrier. You put like a X. A X means they're mating. So these two going to mate. The follow-up line will be the symbols, the parent genotype. So now you have to use your knowledge on how you're going to put the symbols for carrier. Now, if they give you A for normal, S for sickle cell, what would be the carrier genotype?
ES.
Very good. AS mating with AS. To do your Punnett square, now Punnett square is one way you could work it out. You just simply draw like a tic-tac-toe box. This don't have to be in ruler in the exam. You put your parents on the top and the side. So you put capital A, make sure you form your letters properly. Capital S. Capital A, capital. Doesn't matter male and female here. Don't matter that. Now the rule is you multiply the top by the side. So A multiplied by A, you get 2 A's. A now have to multiply with S. So you get AS. Now you're doing top hand side on the next side. A, now even though you say S by A, you always remember you put an A in front. You always put the normal allele in front. So you put A, S, and then you have S by S. Right? So this is the working. Now look at it carefully. You had 1, 2, 3, 4 blank spots here. So it's 4 chances. That means 4 chances that if these two parents were to mate, these are the 4 chances what the child can inherit. So you have one chance at being normal, the child being normal. You have 2 chances, you'll see in these 2, being carrier. And then you have, so I've just put in like a little ratio here. So 1 chance at being normal, 2 chances at being carriers, and 1 chance the child could born with the sickle cell disease. Can everyone see that?
Yes, miss.
Yes, I know it's almost lunch. Okay, I know we will pause in a bit. So if you have to represent a percentage, this will be, so this would have been like 1 out of 4 chances. 2 out of 2 chances, so that is 1. You just put, okay, 1 chance. And this is a quarter chance. 2 out of 4, sorry, that is half. Like half of, right? I just showing you various ways to put the chances. A quarter is 25%. It's always good to use percentages. A half is 50%. And then a quarter again is 25%. So you go back to the question now, and then what they want to know, what is the chance that the child born to them will suffer from sickle cell? So from the results, 25% chance the child can born with sickle cell anemia if their both parents are carriers. That will be like a follow-up line in your working. Because if you look at the question paper, look at how much lines. So you must have some way of stating that, you must state that part to answer the question. If you just stop up to the Punnett square, you done lost 2 marks. You will only get 3. So don't forget that. And that is basically, so that is the Punnett square. If you were to do the, let's say the test cross, that's another way. I'll just show you very quickly before we go for break. You just simply put in AS multiply by AS. And then you're taking your pencil, you're connecting everything. You're multiplying everything on the left, everything on the right. So let's start with the A's. This will multiply with this. Say put down your product one time. A by A, you get 2A. Then you multiply this one now by S, you get AS. And you do the same now. So this S connecting with this A, you get A, S. And then again, so you will notice the same thing. This is the test cross method. The one with the tic-tac-toe box, that is Punnett square. So whichever one you feel comfortable, I always recommend the Punnett square because it's clear. The lines might be confused. It don't have nothing like lines and stuff. If you're ever doing test cross, as soon as you multiply the two alleles, put down the product one time. Don't go and connect all the lines and then you now with your glasses could barely see what you're drawing. All right, so that's it there. For now, we have to pause again. When we come back, we're going back through the last topic. Now, I want to see, like I said, I need to see you doing some questions too. The last topic is menstrual cycle, birth control, STIs. All right, so the menstrual cycle will take the longest there. And then we will jump into some paper ones. So any questions before we go for lunch?
No,
No,
miss.
miss.
All right, we will return. You can log back. You could come off if you want. You can come off. I would give your device, make sure it charge, make sure it get a little rest. You can come off and rejoin at 1 p.m. All right, take care. Enjoy your lunch.
Same to you, miss.
Same to you, miss.
Yeah.
Thanks.